Let me throw a word at you. Protein. What do we think of when we think of protein? Protein powder, protein bar, or maybe you are wondering, did I have enough protein today? Proteins are these molecules that make up big muscles in our body and are itself made of tiny Lego-like blocks called amino acids. We have 20 protein amino acids. That means all of our proteins are made from the same 20 amino acids. However, there are way more than 20 amino acids in nature. So when we eat fish or tofu or any food, we are eating way more than 20 amino acids. We currently know of around 200 of these amino acids, and they have both positive and negative impacts on our bodies. These amino acids are used to treat diseases like Parkinson's or schizophrenia. They are present in our body and make happy hormones like dopamine. One of the negative impacts is that they can act as toxins. For example, an amino acid known as domoic acid can contaminate seafood. So when we eat contaminated seafood, it can cause brain seizures and even death. We don't know how many of these amino acids there are. We don't know what their function is for most of them. That's because we do not have techniques to study these amino acids. That's where my research comes in. My thesis lays the groundwork and asks the question, how many amino acids there are? To do so, I am developing techniques to extract amino acids, to separate them from mixtures and from each other, to identify them and to measure their quantities. For my first step, I used a database called PubChem, which has all the chemicals known to humans. I used that to extract amino acids. I found over 8,000 unique amino acids, from where I created a database called Amino Acid Database. This database will be publicly available. Currently, I am validating this database to identify the sources of amino acids. I will be using this database along with the techniques that I'm developing to look for amino acids in plants, which has applications in agriculture. For example, we can find amino acids to protect our crops against climate change. These techniques can also be used to identify amino acids in all different types of samples, including humans. Eventually, these techniques would lead into diagnostic and screening tests to identify the nutritional and health status of people and plants. Thank you.